Welcome to our second video lecture of IB Biology. Remember that you want to be taking some notes because we get to use notes on quizzes in class. Today's topic, we're actually going to finish up a little bit of topic 1.1, which is just like cell theory. We're going to talk about microscopy, and then we're going to look at the ultrastructure of cells. So not just cells have parts, but cells have so many parts. And those are our topics for today. So the essential understanding, the big idea that we're going to come away with is that eukaryotes have a much more complex cell structure than prokaryotes. Remember that eukaryotes, U rhymes with do and new. So eukaryotes do have a nucleus. Like us, we are eukaryotes, as are protists, those single-celled organisms, like amoeba and stuff, protists and fungi, plants, and animals are all eukaryotes. We have a much more complex cell structure, just lots more pieces and parts than do prokaryotes. Pro rhymes with no because there's no nucleus. These guys are bacteria. So here we have a typical animal cell. Here we have a very typical plant cell. And then down here we've got a prokaryotic cell, a bacteria. And you can see it's a lot more complex. There's just like all these pieces and parts and ripples and bubbles and the bacteria, it's like he's got a cell wall and then he's just kind of gray. And a tail, that's interesting. So definitely still parts, just not quite as many pieces and parts, not as complex a structure. Before we get going, here are our objectives for today's lecture. We are going to talk about advantages and disadvantages of different kinds of microscopes, light and electron. Light is what we have in the lab. It's what we're going to use in the lab. Electron microscopes are definitely out of our budget, but they're super cool. And we'll look at lots of pictures from electron microscopes. We're going to practice calculating magnification. We are going to compare and contrast pro and U. You already did this a little bit with our first day Venn diagrams. So this should be a quick and easy review. We're going to talk about some of the structures in prokaryotic cells and eukaryotic cells. Before we get going too far, here's some of the vocabulary. Much of it you probably already know. If you want to pause the video and think about some of these words, you certainly can. But I'm going to talk about them also in the lecture. So we are going to get ready, get set, go! Here again, eukaryotes, much more complex. U rhymes with do and new because they do have a nucleus. Pro rhymes with no. There's no nucleus in prokaryotes. All right, so microscopes. How do we even know what kind of complex structure eukaryotes have? We have microscopes that help us see them. We have light microscopes. This is what we have in the lab. There's an eyepiece. We literally put our eyeball here. We view through the eyepiece. There's lots of super cool mirrors that bend light. Light comes from here up through our sample. The sample is on a slide on this thing that's called the stage. We have these different objective lenses. These come in different powers, and so we can have a lower power, a higher power. This is the arm of the microscope. This, when we carry it, you grab here. So hand goes here to carry your microscope. We have a couple different knobs. We have coarse and then fine tuning for focusing the slides. And then these guys just help to move the slide so we can kind of push different things into the middle of the view. Light microscopes. This is part of an electron microscope. You can, lots of stuff here, and then lots of monitors, and then computer things, and more computer things, and more computer things. More computer things has to have a special kind of wall covering to keep it all going. These guys are way out of our budget. We do not have electron microscopes at school, but they're super cool. And we'll look at lots of pictures from electron microscopes. So advantages, disadvantages of both of these guys. So one of the big cool things that electron microscopes can do is they have much better resolution than do light microscopes. So resolution, from like your phone camera's pixelation, lots of big squares is less good than more little squares, because this is more squarey and this almost looks round. So we want more pixels to have a better picture. When we're talking about resolution in our microscopes, we're really looking at, can we see between two things that are close to each other? So in a light microscope, if I have these two things that are super close to each other, they might blur and just look like one thing. But in my my electron microscope, it'll be easier for me to differentiate between those 2 points. So that's resolution. In microscopes, light doesn't have a big resolution, just about 0.2 of a micrometer. That's micrometer, sometimes referred to as a micron. It's our shortcut for micrometer. But in our electron microscopes, we can differentiate between 2 points that are 0.001 micrometers away from each other. Our light microscope powers, we can get up to 400 and maybe a little bit more, but it starts getting really hard to focus things at the higher powers in our light microscopes. Whereas in our electron microscopes, we can get up to 10 million times. That's really, really, really zoomed in. And because it's so zoomed in, we can see cell ultrastructure. So not just that there's a cell membrane and chloroplasts, but we can see lots and lots of detail in those chloroplasts in our mitochondria. One of the big benefits of a light microscope, though, is that we can observe living specimens in our light microscopes. We can take some pond water and we can plop it on a slide and we can watch things swim around. In our electron microscopes, we have to kill, freeze, slice, do some other stuff before we can get our samples into the electron microscope. So if you want to see things in color and alive, a light microscope is your better bet. All right. So when we're talking about microscopes, we often talk about magnification, and we do need to be able to calculate magnification for our IB exams. So the formula that we're going to use pretty much for almost all of our magnification problems is this one: actual size times magnification is image size. Which kind of makes sense. If it's actually 1 and I multiply it by 10 times the power, it's going to look 10 times bigger, right? So actual size, how big it really is in real life. Magnification, how much am I blowing it up? And that's going to make it look blown up. So image size, what it looks to my eyeball, because the microscope made it look bigger. So this one, an E. coli cell, has a length of 1.8 micrometers. This is its real size. It is really actually 1.8 micrometers. If I put it under 10,000 times magnification, well, my actual size, 1.8 micrometers. Times, I'm making it 10,000 times bigger. I'm going to have 1.8 times 10,000. That is 18,000 micrometers. You're like, how even big is a micrometer? Is 18,000 a lot or not? So what we can do is convert these into some more commonly used units. So 1 millimeter is equal to 1,000 micrometers. So if I have 18,000 micrometers, I can divide by 1,000, and that's going to be 18 millimeters. 1 centimeter is equal to 10 millimeters. And so I could divide by 10 one more time, and this would be equal to about 1.8 centimeters, which is probably maybe the width of one of your fingers. So it's going to look a lot bigger than it really is. These conversions, you want to memorize these. 1 centimeter is equal to 10 millimeters. 1 millimeter is equal to 1,000 micrometers. All right, let's practice a little bit. So these are all pictures of bacteria. We have bacteria that come in different shapes, which is pretty cool. So these are called cocci. These are bacillus or rod-shaped bacteria. E. coli is a super example of bacillus. These are spirally or spiral-shaped bacteria. So we're going to figure out the magnification of each one of these. I need to take the actual size, I'm going to use my image size, and I'm going to go backwards, do some algebra to find magnification. Actual size is given to me in my scale bars. So these scale bars are telling us the actual size of the image. I just used the word image. not what it looks like, how big it really is. What I'm going to do is I'm going to take a ruler and I'm going to be like, oh, it looks like it's actually bigger because of the microscope. And so then I can do some algebra to find out magnification. This is going to make a whole lot more sense when we actually add our rulers. So again, this is the scale bar telling us our actual size. So actual size is going to be 200 micrometers. Magnification is what I don't know. It's what we're going to figure out. We're going to erase that scribble so we can see just how big our scale bar is. And it looks like it's just about 1 centimeter. So I'm going to put in, for my image size, 1 centimeter, because this is how big it looks with my ruler. This is how big it really is because my scale bar says so. So now I just need to divide both sides by 200 to calculate my magnification. Yes? No, our units don't match. So remember that 1 centimeter is equal to 10 millimeters, and then 1 millimeter is equal to 1,000 micrometers. So I have 1 centimeter, 10 millimeters, 10,000 micrometers. Now that I have matching units, I'm going to put that in a different color so we can see it a little bit better. 10,000 micrometers. So now that I have matching units, I can go ahead and divide both sides by 200 micrometers. Cancels out over here. Divide by 200 micrometers. Micrometers is going to cancel out. Magnification does not have a unit. Our zeros cancel out. So now I have 100 divided by 2. Magnification is going to equal 50 times bigger. So this guy, 50x, is its magnification. Let's do another one. I'm going to make some space so that we can do some more math. I need a bigger eraser. All right. The next one. Our scale bar, how big it actually is 2 micrometers. So I'm going to plug that in for 2 micrometers. Times magnification is what I don't know yet. We're going to set it equal to our image size. Image size is how big it looks on my ruler. And it looks like I've got 3.5 centimeters. Remember that 1 centimeter is equal to 10 millimeters. So 3.5 centimeters is 35 millimeters. 1,000 micrometers in a millimeter. So I've got 35,000 micrometers. To solve for magnification, I'm going to divide both sides by 2 micrometers. Divide by 2 micrometers. 2 micrometers cancels out on the left. On the right, micrometers cancels out. I have 35,000 divided by 2. Magnification is going to be equal to 17,500 times bigger. This one is zoomed in a lot. Good. Good. All right. Pause the video. Do this last one on your own. Restart the video and check your answer. All right, let's do it. Actual size, 500 micrometers times magnification is going to equal my image size, how big it looks. And it looks like it's just about 1.5 centimeters, which is going to be 15 millimeters, which is 15,000 micrometers. Divide both sides by 500. Micrometers cancels out on the left. 500 micrometers. Micrometers cancels out. My zeros cancel out. I have 150 divided by 5, which is going to be 30. Good? Good. All right. So let's talk about some of the structures that we can see because our electron microscopes let us see prokaryotic cell structure. So they don't have any compartments on the inside. They do have a cell membrane. They do have a cell membrane, but that is the only membrane in the whole entire thing. They do have some cytoplasm, so do we. They have ribosomes, so do we. Kind of a cool thing about bacteria cells, though, their ribosomes are a little bit bigger. Smaller. Their ribosomes are smaller than ours. Ours are bigger. Theirs are smaller. We say that theirs are 70S and ours are 80S. And the S stands for Svedgard units. Basically, there was a scientist named Svedgard, and he put some stuff in centrifuges, which are these things that spin really fast. And so there were some things that sank in the test tube. The more dense they are, the more they sink. And so he gave the depth at which these things sank in the test tube, he gave them values. And so 70S is how far the ribosomes of bacteria sank. 80S is how far the ribosomes of eukaryotes sank. So 70S ribosomes a little bit smaller than eukaryotic 80S ribosomes. There are cell walls. Most bacteria have cell walls made of peptidoglycan. It's different from plant cell walls. It's different from fungus cell walls. And then they have a nucleoid, not a nucleus, but a nucleoid. And the nucleoid is simply the part of the cell that has the DNA. There is no membrane surrounding it. It's just where the DNA happens to be. We're going to call where the DNA is the nucleoid. Some bacteria have some super cool little projections called pili. Pili help the bacteria attach to each other. And then they sometimes also have flagella that help them with movement. And then when bacteria divide, they divide by binary fission, not mitosis. Mitosis is all about the nucleus, and if you don't have a nucleus, you cannot do mitosis. Here's another image of prokaryotic cells with a little bit more stuff. So again, we've got that flagella. That's all about movement, how the bacteria can swim a little bit. Endospores, capsules, these are just different ways that the bacteria can protect themselves. They do have that cell wall. There's a cell membrane just inside the cell wall. Pili here. Pili are used for attachment, not movement, but attachment. So if bacteria wants to stick to another bacteria or the bacteria wants to stick to a surface, it's going to use pili. Cytoplasm, just like we have cytoplasm ribosomes like we do, but again, these are smaller. They are 70S ribosomes. And then one single circular chromosome somewhere in the cell, and wherever that chromosome happens to be, that spot is called the nucleoid, not the nucleus. One extra cool thing that prokaryotes have, so there's this thing called the plasmid. It's a tiny little circle of DNA, so separate from its main chromosome. And what it can do is actually share these little pieces with other bacteria. And so if this guy happens to have a gene for antibiotic resistance, then he can make a copy of it. The pili can help this bacteria stick to another bacteria, and then this plasmid can be transferred to this guy, and now this guy also has is antibiotic resistant, which is bad for us. So sometimes you might be asked to draw and label a prokaryotic cell. What you want to do is start with a cell wall, make it pretty thick, so add some extra lines, color it in pretty good. I'm not a super great artist, so pardon my scribbles. You can add some pili. You can add a flagella or two. You're going to add your nucleoid with that single DNA. You can add some ribosomes. You could draw a cell membrane just inside the cell wall. Let's see if I can do that on the computer. Oh yeah, look at me go. Good. And then of course we want to label those. So here are our pili. Whoa. Flagellum is one. Flagella is plural. We have a cell wall. We have a cell membrane. Inside the cell membrane, there's some cytoplasm. We've got some 70S ribosomes. And of course, we've got our circular chromosome, which is made of DNA. And wherever that is in the cell is also referred to as the nucleoid. Good, good, good. All right. We're going to flip over to eukaryotic cells now. They have a compartmentalized structure. So inside the cell, there are lots of other little bubbles, and those bubbles have super cool things going on. Some of those bubbles can concentrate metabolites, so we can keep like enzymes in there, or we can keep sodium in there, or we can keep hydrogen peroxide in there. That allows us to keep toxic or dangerous substances away from the rest of the cell. We do actually make a lot of hydrogen peroxide when we carry out metabolism, and this is super dangerous to ourselves. And so if I can keep it in that little bubble, it's much safer. We can have multiple internal conditions. When we talk about photosynthesis and respiration in our next unit, we're going to talk about a lot of places where we have concentration gradients of hydrogen ions. The membranes that are inside the cell help us make those places where we can build up those hydrogen ion concentrations. They're going to divide via mitosis or meiosis, and then also cytokinesis. These guys are all about the nucleus. This is about the rest of the cell. Good? Good. All right. Perfect. So here's an animal cell. Sometimes it can be hard to see, and so I really like to add color. And now it's much easier for us to find some of the pieces and parts of our eukaryotic cells. Of course, animal cells have cell membranes. The job of the membrane is to control what's getting in and out of the cell. We've got cytoplasm, of course. It's going to dissolve a lot of the metabolites, a lot of those molecules that we need to be able to move around the cell. It's also going to just fill up the space and hold on to lots of water for us. Here we've got a mitochondria, powerhouse of the cell. What does that even mean? It means it makes ATP. Here, these red squiggly guys, so much endoplasmic reticulum, ER, endoplasmic reticulum. And a lot of this is rough ER. Rough ER means that there are ribosomes stuck in the membranes of the ER. The ribosomes are going to make some proteins. Those proteins are then going to get chunked into the ER. The ER is going to transport them to this guy known as the Golgi body or Golgi apparatus. The Golgi is then going to take those proteins that were made by the ribosomes and transported by the ER. It's going to modify them a little bit and then pack them up into these little bubbles called vesicles. So we've got some vesicles. And those vesicles will be transported to the edge of the cell, and then the contents will be released outside of the cell. So if this is like a pancreas cell and its job is to make insulin, the insulin will be made by those ribosomes, and the insulin will be transported through the ER to the Golgi. The Golgi will put the insulin into those bubbles, those vesicles, and then the insulin will be released from the cell. How does the cell know how to make insulin? Well, of course, we have this double membrane-bound nucleus. Here's our nucleus. And inside the nucleus is our DNA. And so the instructions for making insulin are here in the DNA. Good? Good. All right. Let's look at a plant cell. And again, way better with color. All right, so plant cell, we're going to add a cell wall. Animal cells do not have cell walls. Plants do. In plants, it's made of cellulose. Plant cells have chloroplasts. Animal cells do not. Super cool thing though, plant cells do have mitochondria. Sometimes we oversimplify and we're like, plants have chloroplasts, animals have mitochondria. That's not right. Plants have both because they're super cool. Chloroplasts are going to carry out photosynthesis, and then mitochondria are going to carry out cellular respiration. They're going to make the ATP from the glucose that was made by the chloroplasts. One other super cool thing that plant cells have that animal cells do not have, this is a central vacuole It's large, and so sometimes we literally call it the large central vacuole. And this is just going to store oodles and oodles of water. Just inside the cell wall, there is a cell membrane, just like in animal cells. There's a nucleus. And this particular view of the plant cell is a little bit better than our animal cell view because we can also see the nucleolus. So the animal cell also has a nucleolus. It's just that like we had to slice the cell in order to be able to take the picture of it. And the slice that we took just didn't happen to have where the nucleolus was, right? So the nucleolus is here. The job of the nucleolus is to make RNA, to make ribosomes. So this is where we make the ribosomes, so the ribosomes can make the proteins can get transported through the ER. This cell does also have ER. It's just really hard to see in this particular slice. And then, of course, the ER will transport the proteins to the Golgi, and then stuff will get sent out of the cell. Animal cells have a lot more Golgi than do plants. All right, so here's some more zoomed in pictures of some organelles. This guy, anybody have a guess? It's the nucleus. And then again, here is our nucleolus. What I love about this picture is you can see that the nuclear membrane is actually a double membrane. We have two membranes surrounding the nucleus. We've also got some mitochondria here, and then lots and lots and lots of ER and some Golgi over here. Golgi looks a lot like ER. It's just more folded. Good. All right. Speaking of ER, that's exactly what we've got here. Lots of rough ER. Rough ER has ribosomes stuck to it. These ribosomes are going to make proteins. Those proteins are going to be transported through the ER to wherever they need to go. Often it's going to be the Golgi. Down here, everybody's favorite powerhouse. It's the mitochondria. It's mitochondrion is singular, mitochondria is plural. Super cool thing. This guy has a double membrane. There's a membrane on the outside. There's a membrane on the inside. And the inside membrane does all of these folds. And all of the proteins and enzymes that help to make ATP are embedded in that inner membrane. Over here, we've got a chloroplast. This guy also has a double membrane, but its double membrane is just outside. The inner membrane doesn't do any curly stuff. He's got these, looks like stacks of pancakes. This is thylakoid membrane. We're going to talk a lot about this when we get to our unit on metabolism. All right, a few more organelles to look at. Lysosomes are amazing. So lysosomes contain digestive enzymes. This mitochondria, that should actually be singular, mitochondrion, is either dead or sick or dying or old or something, and the body has decided that it needs to be recycled. And so what happens is the lysosome's membrane is going to merge with the mitochondria's membrane. And then the enzymes that are here in the lysosome are going to go into the mitochondria and literally take it apart. Then all of the pieces of the mitochondria can be used for making more mitochondria or other stuff. So lysosomes are digestive enzyme bubbles. Good. All right. Over here, we've got more ER, but these guys don't have any ribosomes. This is known as smooth ER. The job of smooth ER is to make more lipids, more membranes. So it's a lipid manufacturer. Down here, we've got the Golgi. The job of the Golgi bodies, the Golgi apparatus, is to take proteins that were made by ribosomes and then transported by the rough ER, transported to the Golgi. And the Golgi is going to pack those proteins into these bubbles that are called vesicles. And those vesicles are going to be exported out of the cell. So you can see how this guy's getting super close. You can see this guy has actually started to merge with the cell membrane. And so all the proteins that are in the vesicle are going to be released to the outside of the cell. You can see that happening here. This was a vesicle merged with the cell membrane, and out goes all that stuff. So cool. This process is called exocytosis because stuff is exiting the cell. Over here, we've got more ribosomes. Ribosomes make protein. Cool thing, these ribosomes are not stuck to rough ER. Instead, they are free. The other kinds of ribosomes are called bound ribosomes. These are free ribosomes. We have bound ribosomes on the rough ER. We have free ribosomes floating around in the cytoplasm. Both of these guys make proteins, but the proteins that are made by the free ribosomes stay in the cell. So if this cell needs to make some more DNA polymerase so that it can do DNA replication, free ribosomes are going to make those proteins. If the cell is making some insulin that needs to be released out of the cell, then the bound ribosomes that are stuck to the rough ER will do that job. So you've got free ribosomes and bound ribosomes. Good, good. All right, so let's practice labeling some cells. This is IB's favorite animal cell, the exocrine gland cell. Exocrine, like endocrine, but it's going to make stuff that gets secreted out. We've got our nucleus. We've got our nuclear membrane. Here we can see the nucleolus, which is going to make ribosomes and RNA. We've got lots and lots of ER over here. Looks like there's some bubbles of Golgi over here. Let's see, some mitochondria, some more mitochondria, and then, oh my goodness, so many vesicles. This guy is pumping out a lot of stuff. Probably pumping it out into this duct that's going to carry whatever those proteins are to where they need to go. Let's look at this Palisade mesophyll. This is IB's favorite plant cell. We've got cell wall and, of course, cell membrane just on the inside of it. We've got our chloroplasts. Looks like maybe the bottom little piece of nucleus here. Looks like maybe some tiny little mitochondria here. And this big space in the middle is the large central vacuole. And its job is to store water and minerals that are necessary for the plant to survive. Sometimes you're going to have to draw an animal cell. Start with a nice blobby kind of cell membrane. Inside is some cytoplasm. You've got a nucleus with the nucleolus. You've got some rough ER. It's rough because it's got ribosomes on it. You could also have some free ribosomes floating around. We've got some mitochondria. We might have some Golgi with some vesicles. And whatever else you want to put in there. Well, I mean, that belongs in there. Sometimes you'll be asked to draw a plant cell. Make it a little bit more square. Show the cell wall pretty thick. You can show the cell membrane just inside the cell wall. Whoa, I'm not doing a very good job of staying close to the cell wall. But you know what I mean, right? So we've got a cell membrane. We've got some cytoplasm. We've got a large central vacuole. We've got a nucleus with a nucleolus. We've got some chloroplasts with stacks of thylakoid pancake. We've got some mitochondria. We've got some rough ER. And maybe some free ribosomes. Good? Good. All right, so we have come to the end of our lecture, just making sure that we accomplished what we needed to accomplish. We talked about advantages and disadvantages of light and electron microscopes. Electron microscopes can see things way more zoomed in, but light microscopes can see things that are alive. We learned how to calculate magnification. Our formula is actual size times magnification is going to give us the image size. We're going to practice this a lot in the lab. Compared and contrast prokaryotic cells and eukaryotic cells. Pro rhymes with no nucleus. Eu rhymes with do, have a nucleus. We talked about some of the structures that are in prokaryotes and eukaryotes. And that, my friends, is the end of today's lecture. Good work.